Welcome to Health Professional Radio. I'm your host, Neil Howard. Glad that you could join us once again for another segment. In this segment, we're going to have a conversation with Dr. Chris Airriess. He's CEO of CorTechs Labs, and he's joining us on the program to talk about the changing landscape of neuroimaging and the software analytics created to optimize it. Welcome to the program, Dr. Airriess.
Thanks very much, Neil. Good to be here.
A brief background for our listeners who aren't familiar with you. What is your area of expertise? I know that we're talking about brain imaging today. Are you an imaging specialist?
I'm a PhD in neuroscience. Had some postdoctoral work in that area, and then was on faculty at UC San Diego, where I was the director of scientific computing for an international study called the Biomedical Informatics Research Network. We brought a lot of imaging together with high-performance computing, advanced networking techniques, and so on to try and share the results generated from pioneering research centers around the country like Harvard and Columbia and UCSF and UCSD and so on. We're trying to find a way for the people that are collecting MRI images and studies that are very expensive to share the data with other researchers around the country so that we could, basically mine more data out of any given study that is being run around the country. So that was my intro to meet the founder of this company, Dr. Anders Dale, who, when he was at Harvard, came up with the technology behind our company, basically. The company is CorTechs Labs. Our flagship product is called NeuroQuant, and it was the first AI-based application to provide automatic segmentation, which is essentially identification of all of the different regions of the human brain, and then track those compared to where they should be within the normal population based on age and sex of the patient.
Is this technology expanding on previous technology, or is this something brand new that we haven't seen in neuroimaging thus far?
At the time that we launched our first generation of this product, it was the first software available for clinical use in this space. There had been free tools invented, among them FreeSurfer, which is out of Harvard, MGH, and that was invented by our founder, Anders Dale. And so back at that time, he realized that it would be very, very powerful to take that type of tool out of the research lab where you need a PhD to run it, and it takes 6 to 20 hours to run the software, redevelop it sort of around the same core premise, the same core technology, but implement it in a way that is fast, fully automated, and suitable for use in routine clinical practice. And so that's what was done from sort of the beginning part of 2000 through 2006. And by 2006, we had our FDA clearance for NeuroQuant and actively started marketing in 2007 as the only solution for this type of volumetric imaging.
Now, the imaging that we're going to talk about today, is this something that is used across the board in neuroimaging, or is this something that is targeting specific types of neurological problems?
That's a great question, Neil. So when we first started out, the main focus of this software application was to help diagnose Alzheimer's disease. As you know, Alzheimer's is a very significant and growing problem in our aging population, and we felt that this software would basically help find regions of the brain that are essential to diagnosing Alzheimer's and give physicians the information they need to help determine whether a patient that presents with a mild cognitive complaint of some kind is indeed going down the Alzheimer's pathway versus other something else like frontotemporal dementia, vascular dementia, or some of the other types of dementia that can occur that maybe have a better treatment outlook than Alzheimer's itself does. We were also working with drug companies on clinical trials to try to show that their drugs were working. Unfortunately, many of those did not up to this time. But really, the science behind the use of this volumetric imaging was very solid for Alzheimer's disease back in that sort of late 2000s phase. As we moved along, there has become more and more diseases that are showing correlation between changes in brain volumes, changes in the volumes of various structures, to help to diagnose or classify those diseases. So, for instance, multiple sclerosis had for a very long time been considered a disease characterized by plaques on the brain, and these plaques would grow and sort of essentially kill brain tissue and cause loss of function. And so what the diagnostic process was, in addition to clinical assessment, was to look at MRI images, perhaps, and just see whether these lesions or plaques were getting larger or more numerous over time. the scientific literature has come around at this point to show, and our tools have evolved to actually implement that knowledge, such that we now believe that gray matter volume loss, so volume of structures in the deep part of the brain, such as the thalamus, can have a terrific impact on the actual physical presentation of disease, so the amount of disability that a patient faces. So in conjunction with analysis of lesion dynamics, whether lesions or plaques are growing, getting more numerous, or in fact shrinking now if a treatment is being effective, we can help to assess how that disease is progressing and help to provide earlier diagnosis and earlier classification of which type of the disease a particular patient has.
Now, is all of this done through imaging alone, or are you developing algorithms to differentiate between, I guess, certain resolutions or whatnot? I'm not sure how it actually separates the subtle nuances of this type of disease.
We take the images from the MRI scanner, so this is all based on MRI. The patient would go in for a fairly routine MRI scan. We do prefer that they have some high-resolution images for us. They send those images to our software, and the software automatically calculates, basically registers the brain into an atlas space to allow the location of the structures of interest within the brain, such as the hippocampus, the amygdala, thalamus, and so on. It then compares those structure volumes to a normative data set that has the normal range that you would expect to see for a given age and sex of patient, and can tell you where that patient falls with respect to those norms. So you can get an idea if there's a statistically significant differentiation of that patient compared to where they should be for the brain structures of interest. And again, those are disease specific. But again, using the multiple sclerosis example, you'd be looking at thalamus potentially, third ventricle around it, and then lesion burden in quite a few different areas of the brain. So this is something that really assists the doctors in early diagnosis. If you're aware that multiple sclerosis is often not diagnosed or misdiagnosed for several years prior to arriving at a correct clinical diagnosis, this can have a huge impact on the lives of the people suffering from this illness. In addition to finding which or whether the patient is actually suffering from multiple sclerosis versus some other indication, this software can help to differentiate between different types of MS. So, for instance, between primary progressive MS or relapsing remitting MS. And really, we're just working on the AI algorithms to help with that distinction. But what we can do right now is tell you whether a medication is helping the patient, whether their volume loss in the brain is being slowed or even arrested due to the treatment that particular individual is going through, and potentially also let a physician know whether or not the treatment is working as planned, and perhaps provide some evidence that says maybe the treatment choice should be reevaluated if the patient isn't responding as significantly as one would expect from a given treatment.
As far as the development of this technology, what do you anticipate the next phase to be?
We're developing a number of new modules to go with the system. So getting back to Alzheimer's disease, we're now finalizing and about to submit to FDA for regulatory clearance a tool called PET-Quant, which allows us to, in addition to the MRI volumetric information that we have, it allows us to quantify amyloid tracer binding in the patient's own brain. So we essentially register the PET image from a PET scanner into that neuroquant segmentation, and we can tell you exactly which regions of the brain have the most amyloid PET deposition. That, combined with clinical data and with genetics, can give an extremely accurate early diagnosis of patients who are going to be moving towards the Alzheimer's stage if they've got a memory complaint. So getting better diagnostic tools and better prognostic tools that perhaps help patients understand what their likelihood of developing a particular disease is and how long it might take for that to happen is one of the goals of what we're working on right now.
Chris, where can our listeners go online and get some more information about CorTechs Labs?
Our website is www.CorTechsLabs.com. That's CorTechs Labs.com.
Thanks so much for joining us here on Health Professional Radio.
Thank you very much for speaking with me today, Neil.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Dr. Chris Airriess, CEO of CorTechs Labs. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download at SoundCloud, and be sure and subscribe to our YouTube channel at youtube.com, Health Professional Radio.